So, my name is indeed Sam Viersma, and I'm next door in this nice color building at the VUC campus. And I'm a researcher, postdoc in the bioinformatics lab, which is part of the Cancer Center Amsterdam, for box. And we are also part of the Department of Medical Oncology. So, what we do is proteomics, and we do it exclusively by mass spectrometry. So, just to introduce the word proteomics. So, obviously it's derived from genomics. The proteome is the entire set of expressed proteins expressed by a genome, a cell, a tissue, an organism at a certain time. So it's not like a genome at all, or like a transcriptome. It's a function of time and the origin of the material. And obviously it's a blend of the words protein and the word genome. And then proteomics is obviously the study of the proteome. And in 2014, two back-to-back papers were published in Nature stating that we now have final, no, not a final, but a first draft of the human proteome. At that time, the number of identified proteins that are bona fide identified in the human tissue was about 19,000. I think that's shrunken a bit to about 18,000 now, which is still a little bit less than the one gene, one protein number that's derived from the human genome. But at least this is a starting point, a benchmark that we're working on. And what you can do with proteomics is obviously look at differential protein expression. So which proteins are up and down, or down in the case versus control, or as a function of the stimulus. That's more or less like what you would get from transcriptomics. And obviously those things, those methods are interchanged and compared. There's actually not that much overlap between the two, between mRNA and protein. But there's also a set of applications that are proteome unique. And one of them is protein translational modifications, b4tms in short, protein phosphorylation, glycosylation, acylation, ubiquitination. And these are modifications of proteins that influence function and which can only be studied at the protein level. There's no genomics method that can infer b4tms at all. And obviously, for instance, phosphorylation is the key step in signal transduction, interstellar signal transduction, and therefore a key step in, for instance, cancer processes. Okay, the next step is proteogenomics, combining sample-specific mRNA expression and protein expression. And another application field is to find protein-protein interactions. Which proteins are part of a functional complex that execute a specific function. Okay, so that's what proteomics is and what it's about. Colleague Menes will next Tuesday talk about the biological applications of proteomics, and she's also the head of our research group. Okay, so this is basically what I'm going to explain to you today. It's called discovery or shotgun proteomics, obviously mass spectrometry based. I will not talk about antibody based methods. And this is the most generic proteomics workflow that's around. 90% of all proteomics papers that you'll ever read will implicitly use this workflow. You start with your material of origin, cells, tissues, biofluids. Then you extract the protein fraction, the complete protein fraction of this starting material. And then, obviously, the word proteomics implies that we're looking at proteins. That's not what we're measuring. We don't measure proteins at all. What we measure is peptides, so parts of the protein sequence. So the first thing we do is we take a protein mixture and digest it into peptides. Not randomly, quite specifically cell peptides. These peptides can be, need to be separated in some kind, in different dimensions. And the one dimension that you would always do just before the mass spectrometer is liquid phase, reverse phase chromatography. And at the end of the separation step, peptides are converted into peptide ions in the gas phase. Then they are injected in the mass spectrometer, and the mass spectrometer will measure intact peptide masses as well as a fragmentation spectrum. And I will show you what it is. These two spectra are input for a search engine, and from these spectra, the search engine can infer peptide sequences, and these peptide sequences can then be mapped to protein sequences. So the first step is you start with a protein mixture. You can separate a protein on a SGS phage gel, classic 1970s technology, still works. And you can cut out these bands, incubate it with trypsin, that's our workhorse protease that will cut the protein into peptides. Obviously it's a protein enzyme itself. And this intact protein will then be digested into trypsin peptides. There are a few things that need to be optimized. So one is that trypsin is of course an enzyme, so it has quite a big mass. And in order to access the protein of interest that needs to be digested, the protein needs to be unfolded. So what you need to unfold the protein is to add, for instance, a hard soap, like SDS, sodium glycerol sulfate, add a bit of temperature, or put in a chaotrope. A chaotrope is a molecule that will break the hydrogen bonds that form the three-dimensional structure of a functional protein. So this nicely folded three-dimensional structure is then unfolded into a coiled kind of structure, and there's one bond that still needs to be taken care of, and that's the cysteine bond. So the only amino acid that can form intramolecular bonds are cysteines as bridges, and these can be reduced by using, for instance, b4tm or beta-mercaptoethanol, and then the bond is reduced to free cysteines. These free cysteines then react away with the other acetamide, and therefore the three-dimensional structure cannot be folded anymore. You have a coiled, loosely dynamic structure that can be accessed by protease, and then the protease can start cutting. And it doesn't do that in a blind way at all. Trypsin is a highly specialized knife. It will cut only after lysines and arginines. So the lysine is a K, the arginine is an R. And so every word that you think this is blatant nonsense is a peptide sequence. So 20 amino acids exist in nature, and they all have a single letter code, and that's what I will be using throughout the presentation. And they will most of the time end on a K or an R, because trypsin gives you a peptide that ends on a K or an R. Okay, so you can do that for one band, the gel. You can also take a light block or the whole lane. You can also omit the gel and just take your solution with a whole mixture of intracellular proteins. Unfold it by using urea, chaotrope, or guanidinium HCl. Reduce, alkylate, dilute, because trypsin cannot withstand 8 molars of urea. And then trypsin will start cutting in solution all your proteins into nice peptides. And this is a bit of a counterintuitive step, because what you actually do, you start with, we'll say, 10,000 different proteins, and then what you do with the trypsin digestion, you increase the complexity to about 100,000 peptides. Increasing complexity usually doesn't really work, but you get something for free with this increased complexity. That's that the physical chemical properties of peptides are much more homogeneous than of proteins. Proteins are really very diverse in their characteristics. They can be very hydrophobic, small, big. That makes separation steps very hard, physical separation steps. Whereas peptides are much more alike. All these tryptic peptides are in the range of 5 to 25 amino acids, and the range of physical chemical properties is pretty restricted, so that means that a single separation technique can separate them out quite efficiently. That's what we do here. This is the reverse phase separation. Reverse phase is a bit of an old-fashioned term. It's hydrophobic interaction. Bind peptides to a silica bead, which has been modified on its surface with a 18 carbon containing acyl chain. It's like a fatty or a lipid-like surface coating. And if you bind peptides, a whole mixture of peptides at low pH, so put a little bit of formic acid or acetic acid, to these beads, nothing will happen. All peptides will stick to the beads, and unless you start changing conditions, nothing happens. So, and this is a chromatography column. It means that it's a hollow pipe filled with these silica beads, and at the entrance of the pipe, peptides are bound, and during the separation, they separate into individual, or at least separated peaks based on the hydrophobicity. And the only way to do that is to change the hydrophobic environment of the peptides on the beads. And you can change that by, for instance, increasing the organic solvent concentration. So in this case we use acetonitrile, it would be methanol as well. And at the beginning of the gradient, there is all aqueous. So all peptides, irrespective of their sequence and length, will stick to the beads. And when increasing the hydrophobicity of the solvent, for each peptide there will be a concentration of solvent of which the hydrophobicity is equal to the intrinsic hydrophobicity of the peptides. And when the hydrophobicity of the solvent, or concentration of the solvent, increases beyond that point, the peptide will diffuse from the beads and will elute from the column. Physically, this is what it looks like. It's a column of 75 microns in terminal diameter, made of fused silica, and it's filled with particles about 20 to 50 centimeters, and it gives you enough separation capacity to separate out complex mixtures, not in single individual beautifully purified peptides, but at least you simplify the composition at a given time point that elutes from the column. So sequentially, peptides will elute, starting from the hydrophilic peptides and ending at the hydrophobic peptides. At the end of this column, this is only the column, but at the end normally there would be a small connector and a sharp, pulled fused silica needle. And this needle is coated with a conductive coating. And on this conductive coating we have a wire attached, and we put a high voltage, typically between 2 and 3 kilovolt. And that means that at the tip of the needle, between the tip of the needle and the entrance of the mass spectrometer, an electric field of about 2 kilovolt is created. And what happens if you have a solvent slowly pumped through this capillary and then forced through a needle with a very high electric field, what happens then is that a Taylor cone forms. So here you have a kind of a plume, and this plume is because at this tip, at the high potential, the solvent is broken up into droplets. The droplets are charged because we have formic acid or acetic acid, so we have excess protons. So we have positively charged droplets. These droplets are all charged positively, so they will repel each other. So that's why the plume expands. And these droplets float in the direction of the electric field, in the direction of the instrument. At the same time, you also have here a big vacuum system, so also by just air movement, they will move in the direction of the mass spectrometer. And these droplets start shrinking. the solvent, acetonitrile, water, and formic acid start evaporating. And obviously all charges will be at the surface of the droplets. These droplets shrink by evaporation. The charges will not evaporate, the peptides will not evaporate, only the solvents. And that means that charges will accumulate on the surface, and they will be counterbalanced by the surface tension of the droplets. While shrinking, the charge density on the surface decreases until the surface tension of the droplets can compensate anymore. And then the droplet will disintegrate into smaller droplets, and the process will repeat. So, between here and here, typically more than 1.5 millimeter, these droplets will lose their solvent, and at the end, only peptides and excess protons are left. And these protons are then forced more or less on the peptide backbones. And here, at the end of the machine, we've created gas peptide ions. Peptide ions in the gas phase. So we have transferred a liquid solution into a cloud of peptide ions without any solvent. And this is what you need before you put it into a mass spectrometer. Because a mass spectrometer can do a lot of things, but it can only work with gas. It cannot work with liquids. This is the mass spectrometer we are currently using. It's a high resolution, highly accurate instrument. But for a given, this is the layout of the machine, I will show you what the compartments are. But for every mass spectrometer, and there are really, since 1920 something, there are really many designs, mass spectrometers out there, optimized for specific applications. This one works nicely for peptides, but also for small molecules. And they all contain 4 different elements. At least for mass spectrometers, they can do fragment analysis. So what you first need to do is ionization. So that's what I just talked about. Electrospray ionization, that's what I previously showed in the previous slide. But there's also a laser desorption technique, which used to be very popular until like 5 years ago. You need an element of mass separation. So from a mixture of ions, you want to be able to select out one specific ion population. We do that here with quadrupole. You can also do it with a time of flight system or an ion trap. Then you need to be able to hit this selected ion with a method that will induce it to fragment into parts, into smaller fragments. Here it is done in the HCD cell, CID cell. You can also do it in different ways, as long as you smash up your molecule to get structural information, because that's what we do it for. And then these fragments, and in earlier stage also the isolated peptide, need to be detected, mass analyzed. In this machine it's called a polytrap, one of the most counterintuitive mass detectors out there, but it works beautifully. There are also microchannel plate detectors, electromultiplier detectors, and you need this detector to actually do the mass determination itself. This all works in vacuum, so here it's a millibar, and then here in the high vacuum system where the detector works optimally, you're at -10 millibars, but really, really deep vacuum. That's also the reason why these machines make a lot of noise. It's mainly a vacuum cleaning. Okay, so that's a mass spectrometer. And what actually happens in the mass spectrometer is it's a two-trick pony. What it does, it can measure the masses of the intact peptides. It can decide which signal is the highest signal and which is next and next and next. And it will sequentially isolate and fragment each of the peptides that is going into the machine at a given time point. So first we start with a MS1 spectrum, intact peptide ions are detected. Then the machine will ask are there signals above the threshold for detection, the threshold above the noise level? If yes, then we will isolate that peptide ion. We do that in the protocol. And when we have isolated it, we put it in the collision cell, we hit it with nitrogen gas, and we read out the fragment ions. And after that we go with the next, until number 10, and then we start over with a full scan because a new cloud of ions is assembled. And this goes on and on and on during the illusion, the time axis that is created by our chromatography. So we start with a mixture. This mixture is measured, that's the MS1 spectrum, and then sequentially the highest signal, the ion that gives the highest signal is isolated in the quadrupole, fragmented and detected, and then the second highest, the third, the fourth, etc. until number 10, and then back to a full scan. Okay. I can tell that it works, but the manufacturer made a nice move out of that. I've put the volume because the music is quite obvious, quite annoying. So this is the mass spectrometer, this is the inside. We start with the electrospace source here. Ion transfer optics. Here we go around the corner. This means that neutrals go straight ahead and ions can be bent across the ion trajectory. Here we have quadrupole for mass selection. Some more ion optics to get everything right. Here we have the collision cell, and here we have the actual collision. So first we're going to measure the intake mass. Different colors are different masses. So here we have all different ions with different masses going into the machine. You see some ion focusing before it actually gets into the ion optics. Then neutrals go straight ahead, ions go around the corner or force around the corner. Then we go through the quadrupole. There's no selection now because we want to measure the full scan. So we collect all peptides. Then they go into the C-trap here, where they collide with a very low pressure of nitrogen to reduce their kinetic energy, to make a nicely homogeneous cloud of ions, and then they're pulsed into the orbit trap. In the orbit trap, you will see that you get a rotating and an oscillating behavior. And this oscillation is dependent on 1 to the square root of the mass. So therefore, the mass can be calculated for each of these rings of ions. A time-dependent signal is detected, and that's fast Fourier transformed into a mass spectrum. Now we're going to do mass selection, so you have to now watch for the orange Smartie, because that's the one that we're going to analyze. So here we go again with the whole cloud of different ions. You go in, okay, around the corner, and now only the orange ion has a stable trajectory, and all the other masses are kicked out. That's the mass selector. Now it goes into the collision cell. In the collision cell, it's accelerated into a cloud of nitrogen gas. This will induce collisions. These collisions result in fragments. So we've now converted the orange one into smaller, colored, different mass fragments. And these can be analyzed in the same way as the peptide ions. So they're pulsed into the orbit trap detector, start rotating and oscillating, and then the oscillation is picked up by a differential amplifier on the two sides of the detector. And this periodic signal is then Fourier transformed into a mass spectrum. And that's in the end the spectrum that we're going to work with. Other questions up to now? So what you just saw is that first we measure the full spectrum, the intake mass spectrum, the MS1 spectrum. That defines which peptides can be selected for fragmentation, and then sequentially each of the different ions with different mass or different M over Z value are selected, fragmented, and detected. So for each intake mass peak, we have a collision spectrum or a fragmentation spectrum. And both of these you need for identification of the peptide sequence. Okay, so peptides have different forms in which they appear. So what we measure is not mass. What we measure is mass of a charge. So mass divided by the number of positive charges that are on the peptide. And that's because during the ionization process, the electrospray process, we force these charges on the peptide, or the other way around, the peptides compete for charge during the upspray process. And only the charged peptides are actually observed. The ones that are not charged lose before they actually get into the mass spectrometer. So, if you have a peptide of mass 1000 daltons, small peptide, 8 to 9 amino acids, then either 2 or 3 protons can attach to this sequence, to this peptide. And when 2 protons attach to this peptide, then what we actually observe is n plus 2 protons, so that's 1002 daltons, divided by 2 charges. So we see this peptide as at m over z 501. At the same time, if we see the same peptide with 3 charges, it will appear as 1003 divided by 3 is 334.33 dalton. And in your spectrum, even though you put in a pure peptide, you will see it in 2 different forms. Maybe 3 different forms with this large peptide. It will also take up 4 charges. This is a bit confusing. The analysis software takes care of this, but it's good to remind yourself that a peptide can appear in different shapes in the mass spectrum. The good thing is that this leaves a trace that we can actually use, because when we would zoom in, well, especially on this guy, if we zoom in, it will look like this. You will not see a single peak, but you will see a peak with satellite peaks. And these satellite peaks are because carbon is not 100% C12 carbon in nature, but at 1.1% C13. And when your molecule gets big enough, this C13 component, or the 1.1% actually, starts kicking in and start influencing the way it appears in the mass spectrum. And then the strange thing is that if you then zoom in onto the mass spacing, it's a regular spacing between these isotope peaks, it's actually not 1 dalton at all. It's either half a dalton, or 0.3, or 0.33, or 0.25, or 0.2. And that's, of course, if this would be a mass scale, that would be impossible, because there is no half hydrogen mass, not without nuclear fission. So what you see here is actually the Z. So if you see a mass spacing of a half dalton, it means you have 2 charges. If you see mass spacing of 0.333 dalton, it's 3 charges. If you see 0.25, it's 4 charges. So that means that if we zoom in, a high resolution instrument, if you zoom in on these peaks, you can say, oh, this is a 2 plus and this is a 3 plus. Oh, but if I know the charge, I can calculate back what the mass is. It also means that you can, for a single peptide, you can find it different appearance forms and just add those numbers together to actually analyze this peptide. There's different forms, different shapes of the same peptide. Alright, so that's on the intake peptide level. A little bit more real-worldy. This is a HeLa cell lysate. If I zoom in at this time point in the full mass spectrum, you get this. And then if you zoom in on this one, you will see this is the 3 plus form. So, right, yep, the 3 plus form. But then we also see the 2 plus and the 4 plus co-eluting at the same time. Obviously co-eluting, because if it's the same peptide, the peptide molecule eluting from the column, it means that the 2 plus, the 3 plus, the 4 plus, it's all different forms of the same peptide eluting from the column. So there can never be a mass shift between these, no, there can never be a time shift between the elution of these different forms. If it would be a different molecule. It's a new molecule, which might have the same nominal mass. That's possible. But if it's different charge states, this happens after separation, this happens during ionization, different charge states should strictly co-elute. Okay. So we have now looked at interpeptide ions, how they look, what they look like. We can fragment them, and this is the way that works. So we start with the precursor, the intake peptide ion. It's selected, there's no contamination of other ions, so it goes into the collision cell and it hits nitrogen gas. So we get collisions, these collisions will, so we accelerate this, it's not just drifting in, we accelerate it into this cloud of nitrogen gas. these collisions, the energy of the collisions are put into internal energy that makes bonds vibrate and break. And if you look carefully into this tetrapeptide, you could see the regular shape of the backbone. So here we have amino acid residue 1, amino acid side chain 1, 2, 3, 4. Here we have the N-terminus, here we have the C-terminus of the peptide. And if you would break this backbone, there are 3 positions for each amino acid on the backbone where it can break. So we have between the left of the carbonyl, we have between the carbonyl and the amide, and then right of the amide until the next alpha. And the good news is that if you have this CID or HCD cell, low energy collisions, that means that acceleration is tuned to quite a low value, what you induce is a single break in the backbone. Single peptide backbone breaks, that's what the machine is aiming to do. And the weakest bond that's broken is the backbone itself, and it's actually the backbone between the carbonyl and the amide. So only Y and B ions are to be observed, and the X and the Z and the A and the C are not observed under this type of collision cell design. It's important to realize that what you get here is not a single break point. It's not that you break the molecule into 2 halves, but that's like 2 halves. What you do, you make a single break, but this break can be at different positions in the backbone. And it's actually the frequency distribution that will show you, that will give you a very high peak if a single break occurs more often than other breaks. So we are looking at single peptide backbone breaks, but in the real world you also get internal fragments, chemical and electronic noise, you get neutral losses from specific amino acids, and sometimes fragments are missing. That all these steps complicate the invitation or fragmentation ion spectrum. Here to give you an idea of the numbers, these are the molecular weights of the different amino acids. So glycine is the smallest, 56 daltons, and then the biggest one is tryptophan at mass 186. They have different properties, plotted in different colors here. And the trick is to assign the B and Y, or the B and the Y ion peaks to specific amino acids in your sequence to elucidate the peptide amino acid sequence. So my parents were so kind to give me a name in amino acids and even a triptych name. So it ends on R. That is perfect as example. Here you see the B ion, B1, B2, B3, B4, etc. And from the C terminus, the Y1, Y2, Y3, Y4. These are the fragments that you can then observe, and the counterpart on the other side. And for each of these fragments, I can calculate to the 10th decimal what the mass should be. It's always the N-terminus plus residue mass, N-terminus plus 2 residue masses, N-terminus plus 3 residue masses, and here the C-terminus plus 4 residue masses, etc. So from a given sequence, I can calculate all possible B and Y ions from left to right. So this is just a small puzzle where you know the sequence, you can calculate all fragment ions. So if you then have an experimentally measured spectrum, you can assign all of these peaks with the corresponding fragment ion label. In real world, you just get a spectrum without any labels, so that makes life a little bit harder. What we used to do 20 years ago is that you would then start, knowing that you have either a B series or a Y series, you would start measure the distance between peaks, and sometimes when you're lucky, you get exactly an amino acid mass difference. You know, okay, this amino acid is part of the sequence. If you have 2 or 3, then you would know, okay, we have an ER, that's part of my peptide sequence. I can do a blast search in the protein database and see whether there are peptides containing a tripeptide somewhere. That's de novo sequencing. That's hard, tedious. Some people are good at it, but computers are much better at it. Because the computer can cheat. the computer doesn't really try to get mass differences. The computer starts with prior knowledge, because we have the whole genome sequence. So we know what for the whole 20,000 protein sequence, we know what the sequence is at the amino acid level. If you include isoforms, you have 40,000 protein sequences. And what you can do, or what the computer does, is take all these FASTA files, which is just a single header followed by the single letter amino acid code. And for each of the proteins, we start digesting it into triplet peptides after each KMR new peptide starts. So we can predict all peptides that are triptic, that are between 7 and 25 amino acids, and we can calculate the intact mass of each of them. For different charge states, 2, 3, 4, 5. That's all very trivial. We can rank them by mass, or by M over Z actually, not by mass, but M over Z. So we get a very long list of numbers, intact masses, and sequences. If we can go back to our data file, we have an intact peptide mass, we have a discrete intact mass observed for it, we know its charge state, we also know how good a mass spectrometer is. Typically our machine should be better than 5 ppm mass accuracy. So we can take the detected intact mass, go 5 ppm up and down, and that mass window we compare to our list of predicted M over Z values, and we pull out all peptides that fall within this window. For each of those peptides, 100, 200, 300, we calculate the B and Y ions, and then we go to our measured MSMS spectrum from the isolation of this -1 peak, and we compare all predicted mass spectra, MSMS spectra or MS2 spectra with the actual measured one, and then we give a score to each matching fragment ion peak. We give a punishment for all missing peaks, and we aggregate all these scores into a single number. And that means for every sequence we have a score, and we can rank them. And the assumption is that the first one is the right one. And there are different ways of going about this. Does this score have to be above a threshold, or must the difference between the first one and the second one be very large? And until 10 years ago, 5 years ago, there were a lot of in-house rules to value or to validate your peptide identifications. And within a lab that was all okay, but it meant that it was very hard to compare peptide and protein expression analysis between us, because everyone has its own in-house rules to judge the quality of the peptide identifications and protein identifications. This was solved more or less, well, I think it was 8 years ago, by doing false discovery rate calculations. There's no prior assumption on what a score should be, a high score or a low score. What we try to do is explicitly estimate the chance that we have a false positive identification. So what we did until now was to search all the spectra against a sequence database. But that doesn't solve the random identification problem. There will be random matches, you want to know which ones they are, and you want to filter them out. And one way, a very successful way to do this, is to take all peptides that the search engine has found during digestion of the FASTA files, so preparing the search space. And for each peptide, in the forward, the same peptide was taken and reversed. So that means those two databases are identical in size. Size, the properties, distribution, everything is the same. The only thing is you have forward sequences and reverse sequences. Forward and reverse, that means that B and Y ions are different. Those numbers for reversed sequence are completely different from the forward numbers. And by definition, they are false positives. Or they are really false identifications. And then the assumption is every identification from our real set of MS spectra against the reversed database search should be a random match. It's wrong, and it's a random match. And the frequency in which it occurs will be the same for the reversed as well as for the forwards, because it's random. So what we do, we search exactly in the same way as we do here, but now against a database containing all forward and all reverse sequences. The assumption is that reversed identifications will give a very low score because they should not be there. And here actually I've plotted all forward and all reversed hits as a function of the score, the Andromeda score for search engine that we use. And then you see indeed that the reversed hits cluster around low scores, and then the forward hits, they are shifted to the higher scores. And what you can do is just to take this bar and calculate how many are in your forward search and how many are in your reverse search. And then we arbitrarily set a threshold of 1%. So we accept that 1% of all our identifications are with a score which is similar to the reversed score and are most likely random matches. So this threshold can be moved up and down, of course, but typically it will be 1%. And by this, by FDR filtering, it means that different search engines can be compared to each other, and data of different labs can be compared to each other. And that really made the proteomics world much more transparent and much easier to reprocess data. The important thing is to note that, I guess for this audience that's a bit trivial, but that the FDR is a global number. It's not a number for a specific peptide, it's only for the complete data set. And therefore, obviously all the reversed hits are filtered out of the data set up front, but we only know that 1% of our forward hits will be wrong as well. Most likely the one at the lower end, but it's a global number. So finally, in real life this is actually quite important, because what we talk about now is only identification. So which peptides are there, which ones are there in my sample, in my soup. And then we can map peptides to proteins. This is quite straightforward because you already start with a FASTA file for creating a complete sequence. So mapping peptides to proteins is not that hard, although there are some inference rules that can be a bit tricky. But assuming that all went okay, then the next step is how much of this protein is in my sample A and how much is in my sample B. And that's actually the correct question to ask. The wrong question is to ask how much of my peptide A is there compared to my peptide 1, compared to my peptide 2, compared to peptide 3. Because what we can measure in a label-free experiment is the intensity, the peak area under the eluting peak of the MS1 signal. This is a number that can be easily calculated. That's not that hard. But it's a function of the ionization properties of the peptide. So that means that although we have this protein, so we assume these peptides are stoichiometric, 1, 1, 1, the signal on the MS1 signal can be really different because in competition for charge, some peptides are better than other peptides, and they give a higher response on the detector. But it will be consistent. So if we have the same protein, we will observe a similar set of peptides, and if I compare this peptide to this peptide, it's the same peptide coming from the same protein, and these relative numbers do make sense. And if we add up these numbers and add up these numbers, we have a relative value for protein expression. It is very useful. At the same time, there's also an easier way to do this, and that's just to count the number of identified MS-MS spectra, the number of identified MS2 spectra for each peptide that are mapped to this protein sequence. And if you have more than 10 spectra on average, the sum of more than 10 spectra, there's a very high correlation between the sum of the intensities and the sum of the spectra. Only at a very low count rate, this correlation is not that good. But for any decently expressed protein, counting, spectral counting is quite a nice method, pretty straightforward for quantification. Both of them are used in practice, both of them can be used, and both of them can be used, the number that comes out of it, can be used to look at protein regulation or differential expression between samples. So the right question is, a given protein across samples, in different groups for instance, that's the comparison that you can make. Not one protein versus another protein in the same sample, or one protein versus another protein in another sample. Those are useless questions. The same protein across conditions, across samples, that's the right question to ask. And label free quantitation will directly give you that answer. If you really want to know the absolute quantity of this protein, then one way to do that is to synthesize peptides with isotopically labeled standards. So carbon-13 enriched and 15 enriched peptides, they can be synthesized, you can quantify them in a different way, quantitative minor acid analysis, spike them in your samples, and then the relative areas can be used to calculate the absolute amount of a peptide and therefore a protein in the sample. But that would be like 1 to 5% of all proteomics maybe will be about free quantitation. Normally, relative quantitation, label-free is well, that's our own method that we like. There's other labeling strategies that can be used like a differential relative quantitation method. Okay, so to summarize, this is all the steps that I've gone through. A little quantitation here. The important thing is that that you use the right biological context, and especially in selecting the FASTA file. The most common novice proteomics analysis mistake that's been made is that you don't want to miss out on anything. So what you then do is, okay, I'm looking at an experiment with human, well, maybe adding a mouse and a few microbes, because yeah, I need a host infection going on, don't want to miss that. So I just take NCBI non-redundant, all species that I can think of that have been sequenced. At the same time, yeah, maybe there is some signaling going on here, some phosphorylation, glucosination, protein degradation. I don't want to miss out on that at all. So as variable modification, I turn on everything that I can find. Every modification that I can find, I turn it on. What then happens is that you explode your search space, and the chance that you will find anything biologically relevant out of that modification or other organism search is very low. Normally you know if up front, if your sample prep was good to enrich for a specific modification, or if there were other organisms in your sample that were relevant. Typically not. Typically it will be human, or mouse, or yeast. Sometimes, if it's very complex, you have mouse and human PDX models, human tumors in mouse, implanted in mouse. That's already a very hard question. So this is a recipe for failure. What you need to do is stick to the single organism that you think you are working with, unless you have other arguments. And for post-translational modifications, only search for them when you have enriched for those peptides. If I do a phosphor experiment, I actively have to enrich for phospholipeptides because they will only form 1 to 5% of my total signal. It will swamp away and normalize it. But if I do that, then 95% of my peptides are phosphorylated. Then I turn on phosphorylation modification in the search engine, and I will find many phosphorylated peptides. The other thing is proteomics, but that's not what we measure at all. That's what we infer, not what we measure. What we actually measure, the primary observable, is peptide MS-MS spectrum, and the rest is indication. So after a large-scale screen of many different conditions. You have a hit list of candidate proteins. We started with 5,000 proteins, and I'm top 3. And then you will see on number 1 there's always this single protein that nobody ever has written about. So that's really not novel findings. But yeah, of course, this was a protein that was identified with 2 peptides. Borderline score. It's very important if you think you're going to spend half a year on validation with functional or with orthogonal techniques like antibodies. Be sure that you know what the actual evidence is, how good the evidence is. And if there's alternative, if number 2 has been found with 50 peptides straight on very, very strict identification, start with that first. Save the 2 peptide hit for later. So you really have to dig down into primary data to convince yourself that you're really looking at something that's real. Well, the first database search this tends to be a optional thing. It's now completely it will accept your data set without filtering. Quantitation can be done by different methods. Label free or with labeling, intensity, or spectral counting. Every quantitation method has its plus and minuses, pros and cons. As long as you validate it within your own experiment, all of them are valid. And at the end of the day, you're stuck with a list of differential proteins for regression analysis, and you will need statistical tools to see whether those proteins are actually, the fold changes, are actually statistically relevant. And that's what Tom is going to talk about in the lecture. And I think this is it. So any questions?
A question, how do you deal with problems working with human samples? Sometimes in the column gets stuck. How do you avoid to get this problem when analyzing peptide?
Yeah, so that's one of the things that can make an experiment fail. If you have contaminated samples, especially with particles, these columns will clog immediately. So these are HPLC columns. So some people say it's high performance, others say high pressure. This runs typically at 500 bars. So that means that there's a small clock at the entrance. It means everything will jam, and it will just HPLC will stop. Well, there's actually nothing that you can do when it happens, but you can prevent it by pre-treatment of your samples. What we typically do is we do like a preparatory reverse phase step. So we first phase material, the same as in the column that we apply the peptide to, and we elute it as a block. We immediately go from aqueous to 80% acetonitrile. Then you elute all your peptides as a clean solution, and then you evaporate off the acetonitrile and inject peptides that have been cleaned. Another way of doing it is, if you don't want to do solid phase extraction, is that you use a microfilter for all your samples before they actually go into the vial from which the sample is sampled and out of sample. That is typically an Oppo 2 microfilter. That takes care of most of the issues. So this microfilter approach, it took us 5 years to find it. And before that, it was always very hard, because if somewhere in your experiment the column clocks, it means that either you have to repeat everything you've done before that, or you have to, another way, repair the data set. And after that, we've never had a clogging anymore. So getting particles out is important for stability, especially for large-scale experiments. Sounds trivial, but it's
I'm just curious, like the whole procedure, how long will it take to identify the protein?
Oh, well, if it's well, you can't really see it. So for a standard, not too deep proteome, so single shot, I will identify from an experiment of, this will be 2 hour, well, no, it's a 1.5 hour experiment for a single sample, single injection takes 1.5 hours, and it will give you about 3,500 protein identifications, 20,000 peptides, 3,500 protein identifications. If you need to drill deeper the proteome, it means that you need to fractionate. So instead of injecting the whole digest in one go, you can fractionate this. You can do it up front. You take a gel, you put it into 5 different blocks. Then you do in gel digestion, inject 5 times your samples. Then you go to a depth of about 5 to 6,000 proteins. If you would fractionate to 10 fractions, either on a gel or maybe on what you can do is high pH reverse phase, and then you can fractionate in the liquid phase. Then you can go to 8,000 to 9,000 protein identifications. There are also labeling strategies that allow you to multiplex 10 samples in one go. And what you typically do, you fractionate that into 24 samples, and you go to a depth of 10,000 proteins. That's current state-of-the-art living, 10,000 proteins for a given sample. But it goes at the expense of time.
But those 10,000 proteins, you all know their sequences?
Yeah. So you see basically half of the one gene, one protein, one proteome. It really depends on what you're looking for. So our central question is for instance treatment options. People which have increased expression of this and this protein will respond better to this and this drug. So that means that if you drill to the depth of 10,000 proteins, then you also have very low abundant proteins. The chance that they make it into such an assay, if you are at the 3,500 protein range, that's still so. This, if you would see proteins like an iceberg, this is the top, a little bit in the water. Still medium to high abundant proteins. Those are ones that you can reproducibly identify without much problems. So we tend to balance the depth that we need to the final application that we're looking for. So it's a bit of a matter of choice. So sometimes it is the low abundant protein that makes a difference. At 3,500 you will miss it. You might need to go to 10,000 protein levels. But it's a bit of a balance what you can do within the time that you are willing to spend on the experiment. Just brute force will not work. It's clever experiments. It's not brute force. But you still need at least some brute force to drill, to identify and quantify enough proteins with a high enough quality. Other questions?
How many cells did you need to run the experiment?
Oh yeah. Well, I don't really count in cells, because cells tend to be pretty diverse in protein. But for an expression experiment, if I look at if I use a gel method, I would look for a blue lane. That means 50 milligrams on column, about 50 micrograms of protein, more or less. If I want to look at tyrosine phosphorylated peptides, that's 1% of all phosphorylations, and 1% is most interesting, and it's 1% that's most harder to detect. I need to start with 5 to 10 milligrams of protein before I can get to a range of about 1,000 phosphotyrosine peptides. If I want to look at all phosphorylations, serine, threonine, tyrosine, I can measure 10,000 phospholipeptides from 500 micrograms of starting material. It's hard to convert that to number of cells, but it's doable. So from a biopsy, I can get in the order of 1 to 2 milligrams of protein. So for phosphorylation analysis, expression analysis, that's enough, no problem. If I want to do single cell proteomics, there is a small niche field that actually aims at that, following the mRNA world, where you do single cell sequencing. Yeah, it's still a bit of thinking of tricks how to do that, and then reverse, and then calculating back. Yeah, I started with 1 cell, equivalent of 1 cell, but then you need, of course, 1,000 of those cells before you can measure something. It's a bit of cheating. I don't really believe in single cell proteomics, but still, maybe in 5 years' time, term for old-fashioned.
So in this part time, would you see, is it used in the clinic, or let's say you have a tumor and you come and then they take a biopsy?
For diagnostics, it's not used. No, in a experimental setting it is used. So we are doing retrospective studies. So we have tumor biopsy material, pre and on treatment, and then we look whether the targets for the drug are present in the biopsy. But we haven't. We are now gearing up to experiments where we can, where you have like the classical drug selection and the molecularly based drug selection based on, for instance, phosphorylation profiles. But there are up to now no patients yet that have gotten their drugs only because of phosphorylation patterns. So mutation analysis is standard, but the proteomic counterpart of that is being developed, but it's only in the clinical research setting.
So in the interest of time, I think this is a
Yeah.
nice point to cut it off. Let's thank Sandra again for the clearest explanation. Okay, so we are running a little late. I'm not sure if this room is booked after us. If it is, we will run out at 1 sharp. So any questions that aren't answered by then, we'll pick up digitally instead. Before we start, a quick reminder that there is a research project introduction lecture by Anton at 3:30 in N129 of the WN building this afternoon. So that is 3:30 in M129. Ready to pick up where Sander left off is Dr. Van Van, also from the Cancer Center in Amsterdam, right here at the UMC, who will tell us how to actually analyze all the proteomics data that you now know how to measure with LC-MS. So, the floor is yours. Thank you.
So we'll continue with statistical analysis of proteomics data, and as the title said, it's a little bit mathematical, but I know the background of the audience is quite wide, so we will go through some very basic stuff and go quickly through some more elaborate things. And there are papers and slides available, so you need to note down all these mathematical things. So my name is Tang Pa, and this is my email. We always welcome internship students. In the next few lectures, you will get a lecture by Professor Cody Jimenez, head of the lab, and she's a biologist. So we have topics in biology, medicines, and I'm a computer scientist. And I also have a few slides on some of the things that we've been working on, so quite interesting. So if you have interested, email me or call me later. I always draw the parallel between the genomics data and protein data, and we do sort of similar thing, but the technology, the underlying technology is of course different. For sequencing data, you have sequencing machine, and for protein, we have mass spectrometry that Sandra just covered. So today it's more about statistical analysis. So if you have any experience with genomics data analysis, there will be tools like Lima and HR. But as I said, in application, we do things that are very similar, like here is an example where we have a cohort of colorectal cancer patient, and the aim is to group the patient into subtypes, and each subtype might exhibit different characteristics. And we are working on one project where we combine RNA sequencing data and protein data to make colorectal cancer subtype. There are different way of subtyping, but we are working with one system where they classify this patient into 4 groups, CMS 1 to 4, in which CMX4, for example, have a very poor prognosis, and these patients normally do not respond to chemotherapy. So we build classifier to separate this group patient using proteomics data. And of course, other people have done that with genomics data. And one of the things we want to do is also to combine these sorts of variation. Just quick recap to the last lecture. I don't think this needs to be done. So you have iron column here, and it's going to the mass spec. Important point here is that there's two ways, or at least two ways of quantifying a protein. First is one look at the area under the curve of the MS spectrum, and that's the ion intensity method. The other one, which is actually very easy, that for each peptide you count the number of times that it gets sequenced into MS, because the machine tries to break down the peptide that has the highest intensity. So spectral counting tend to approximate ion intensity very well. So that's the advantage, that you have the identity of the peptide and you have the quantification of the peptide as well. For more elaborate signal processing, then you can get the area under the curve of this peptide coming out of the machine quite accurately, and there is, if you want to be very precise, then ion intensity is there. So that already I covered these two parts. Protein identification, we got the last lecture, and also protein abundance of quantification, so how much of this protein is in the sample. And we primarily, in this lecture, focus on the hypothesis testing of how much between group condition. And before we do that, I always want to remind people that data visualization is very important. So, for example, this is a rather old mass spec spectrometry approach, but you see in the axis is the M over Z, and on the y-axis the intensity of the signals. These are the signals that are coming out of the machine. And after doing all the analysis, one can sort of overlay the signal and get a very nice visualization. So, do people know what this isotope pattern means? The last lecture. All right, but this is just the basic of the mass spectrometry. Why do you have this second bit here? Yeah, so that just carbon 1% has to the weight of it more 13 instead of 12. So the last lecture you do a lot of this type data. So in addition to the MS data, you have also the extra separation. And actually we can do similar thing, visualization. So this is one peptide in 3 dimensions. You see that all the peptide around it has similar kind of pattern. But this particular biomarker, this is on real data for healthy people. You see very, very small peak, whereas at that particular location, for the people who have cancer, it's very high. So that's the sort of validation of your analysis. So we come to this part, and who doesn't know p-value? Okay, well, so that is very easy. Just very quickly, so the p-value, we always have this concept of null hypothesis, that data generate from the same distributions, and we want to see if we can reject this null hypothesis, and p-value is the probability of obtaining a test statistic that's more extreme than the one that under the null hypothesis. And because most of you, I guess, is computer scientists, so I would try to explain it with permutation test. So you have a random variable. This is a normal distribution. These are parameters. I just want to repeat some very basic things so that later on we can our lives a bit easier with the conception. We have parameter of the distribution. So for example, a normal distribution with mean of 100 and 1,000 sample, and one can plot a histogram like this. Right? Just stop me if it's too fast or something that's unclear. And I have another distribution of another of the same protein in another 1,000 sample, for example. Here you see the mean is shifted by 10. Okay? And the variation is the same. So if you plot the two together, you get something like this. And one asks if these two conditions are different. The expression of these proteins are different between these two conditions. But if the spread of your data is bigger, this is the same plot. You see it's the same kind of well, the same difference between the mean, but you have a different feeling for the differences. So how can we quantify these differences? So this is the t-test, is the one way of doing it. So this is the mean of your distribution, and this is the variation. What's important here is that the statistic is just a function of the data. So given the data, you get a number. That, for computer scientists, that's very easy to understand because the data is just input to your function. Here I should mention also now that if you think about this set of numbers, if we add a constant number to this data, right? So if you plus add whole number with the same number, what you get is that the mean will be increased by that constant, and as a result, this value doesn't change, and the variation doesn't change. So if you have two groups, and each group added the same constant number, what you notice is that the difference between the two means will remain the same because both are changed by a constant number, and this one is the same also. So the t statistics actually invariant to addition. We'll come back to that. So how to calculate a p-value? So if you have a t-statistic of your data, in two groups, group 1 and group 2, we calculate t0. And then the permutation test would just pull the all sample together and repeatedly divide the data into group of psi n1 and n2 and computes the t-statistic again. And the p-value is a proportional time that this random p-value is more extreme than the one that we observed originally. So I guess the p-value can be very confusing, and some people have difficulty understanding it, but as a procedure, it's very simple. Well, theoretically, it can be, if this is another way of calculating p-value for a small number of samples, and I will not go into this. So if your data is not, let's say, there's assumption behind the t-test, and if your data is not, let's say, normal distributed, this is another test that it can be used to that. It's called the Mann-Whitney U test, and the test is actually quite simple. You take the minimum of these two values, where U1 is the number of times that the value in X1 is bigger than X2. I just asked the group if you know the complexity of calculating this. Any computer what does it take to count the number of times that x1 is greater than x2? Let's say x has n points. Sorry? I think more, because for each value in x, you have to compare to everything in x2, right? Count it. Yeah, can we do better? Yeah. Every time I ask this question in PDC, people got it right actually. Well, if you do, you sort the list first, then it's n log n. Well, of course, now if you do the test, you don't have to implement it anymore, but I just want to make you think of it. And, you know, this kind of test is implemented in R and packages. It's good to just think about it. So now we come to the real thing. So spectral counting, as I said, and you get from previous lecture as well, it's very simple in the sense that for a profiling experiment, for each peptide, you want to know what it is, and that is the identification. And in order to do that, we break the peptide into smaller pieces and then do all sort of database matching or other spectral matching. And the number of times that this particular peptide gets sequenced, it's a proxy for its abundance, and we have counting numbers, integer 0, 1, 2, and so on. And this is an example of a real experiment of cancer and control, and these are protein identification, which is not recognizable, but anyway. So we try to do, how do we find the proteins that are different between these two groups of samples. And these are 6 markers that is known in clinics that these should be differential between the two groups. So the adenoma and colorectal adenomas are the samples that, let's say, pre the cancer stage. And we want to find proteins that are different between the pre-cancer stage and the cancer stage. And we tried the method that was in the review, and we get, for some method, you get sort of p-value, small p-value. So here you expect small p-value because these are markers that are known in practice, so to say. But they are not quite satisfactory actually, because you see p-values are quite large actually. So we try to go back to see what is really going on. And the first thing we do is, of course, a t-test. And if you do the t-test for this, everybody knows R, right? For this two set of samples, 1, 1, 2, and 2, 3, 3, 4, and you get this p-value 1%. And you give this to Sandra, he said, okay, that makes sense. Maybe we should look at the intensity to see if these proteins really changes. And but if you add 100 to this number, so as I said previously, you put in R, you get exactly the same p-value. And this is not good, because to people in the field, these numbers are actually almost identical. And the reason is that if you have, this is then real data again, we have a sample that you run every now and then to control the quality of the machine. And for one particular protein, it has about 30 counts. And these 29 runs out, and these green dots, green triangles, are its value. So even with one biological sample, and you just measure it again and again, you get this kind of variation. So the red lines are, let's say, the variation, the distribution of that sample, and the green are just well, we ignore the green now. So you can imagine that for a protein of about 100 cal, you also get a range of from 80 to 120 if you do it again and again. So that means that we have actually two sources of variation here. Here you have the variation due to the sample being a cancer or pre-cancer. But there's also another source of variation that is called technical variation. What it means is that for this sample, you get 7 cal for this particular protein, but if you just do this again, if we were to measure this sample again, we might not get 7, we might get 3, we might get 5 counts. And same for 48. If we measure it again, we might get 40, we might get 46. So that's actually what it means with the blue line you see previously. And tomorrow, these two variation test this formula that you do not need to understand by now. Well, maybe you do. It's just underlying everything that I will be talking about today, actually. That you have, this one is technical variation, and this part is called the biological variation. And these phi's are, sorry, the theta are the parameters of your distribution. So the normal distribution, we have seen we have the mean and the variation. And here we just have a set of parameters. It could be normal, but it could be other distribution, right? So this integration together give you a the distribution of your data. So the x here is can be cancer and normal, and the y is what we observe with this count. So the first thing we do is that, okay, now we have seen the technical variation because of the 29 control samples that we do. So the sample that we measure again and again, and we see that it can be approximated by binomial distribution. So what is the binomial? So that's the red line that you see in the previous slide, right? The red line and the blue line. So that's the technical variation, the binomial distribution. And the binomial distribution has two parameters, the phi and this total count. Well, it's quite a bit technical. But what is said here is that if you combine these two distributions together, you get a very nice closed form distribution. And what it means is that you can put it into a optimization. It has derivatives, it's a second order derivative. So optimization is very easy. That means that given data, we can estimate the parameters of the distribution quite easily. So the same for a normal distribution. If you have the data, we can calculate the mu and the sigma in closed form. Here, not closed form, but still easy. So this is real data. So again, this is the plot of the binomial distribution that we see in previous slide. And these are the 6 sample points. And as you see, the binomial model does not model well this data set, because there's even 4 points out of 6 lying at extremely low density region here. Whereas this data binomial model represents this 6 data point quite well. And we perform evaluation on this test versus many other tests, and beta binomial is in purple here, performed quite well in terms of false positive rates with the same rate. So it's a nice impression. So in another setting, you have data that coming from single patient that before treatment and after treatment. So these data are we compare data, and we have so it's not independent test as well, and again a little bit elaborate, but sliced variable, and that's also paper, and it's this is the formula that I showed before. Again, the technical model, variation and biological variation combined. Now, this is not like the beta binomial where you have a closed form and optimize and it's easy. This one is quite complicated to optimize, so to say. But this is doable. So given the data, we have this method to estimate the parameter, the distribution, computation. Yeah. And we perform an evaluation against a very popular method at the time, the HR. And as you see here, that for example, the HR method that for HR has a low p-value here. So this part of the Venn diagram. So it's low p-value for HR and not for the inverted beta binomial test. So you see that here, one sample is from 200 to 43, and another is going up from 80 to 150. So for a test that testing for the trend between normal and tumor after treatment, one go down and one go up, and it's still high in HR. So we perform quite extensive evaluation between these two tests, and this test actually being adopted by many people in genomics as well. So one thing then was we noticed that as the machine technology progressed, it gets better, and also with the technician in the lab that do things better and better. So what happened is that the variation for technical variation get better basically. So if you measure something 30, before it's a black curve here, but as the technology get better, you can get much more precise measurement if you just do things again. So the beta, the binomial distribution might not be the best anymore, and we can have this called the Poisson distribution with an extra parameter that can actually vary the technical variation. And so the green dot here means that it's actually more spread than the normal binomial distribution. It is the case where you try new techniques that, like phosphor proteomics, you have to do extra capture, and it's if you do just it's more elaborate. If you do things again and again, you get maybe not as good as the one that you do regularly. So the challenge here is that it should be trying to devise a new method for every new new analysis method for a new technology development, for example. So here you have, for both the data, what we have seen is the DDA data, the LCMS data. Did Sander mention about DIA data? Data independent acquisition. Okay, I think it's very very exciting as well. What we have for data dependent analysis is that for peptide that is for big large protein, you get sequence, and you can always identify the one that are big. But for another kind of measurement, we do not sequence the big one, but we sequence in regular space. So there are different ways to measure the data. And also there's phosphoproteomics. That means that before doing the LCMS, we do a capture of the phosphopeptide and do the mass spectrometry after that. So the challenge is that you have different normalization procedure as well. So if what coming out your equipment actually not car data, but you have to normalize for some analysis method. Different mode of quantification we have said already. Some people prefer intensity instead of count. So the idea is that we want to develop methods that can adapt for different technical variation. So I'll see this one. So I go back to this equation. So if we know the parametric model of our statistical variation, that would be easy. So that's like normal distribution. We have seen before, the mean and the variation, and given the data, we can estimate the parameter. But what if we let the technical variation, let's say, can be freely designed, right? So these are the biological variation, which actually quite safe to assume to be normal because we don't know what would be the difference between normal and cancer or cancer and pre-cancer. So recently we show that if we are willing to make that assumption that the biological variation is a normal distribution, then we have a very efficient method for model fitting, and this can be implemented in 20 lines of topics. So very fast, very efficient, only that it's in MATLAB, and it's difficult for people to use. So we are working on making this in R and also making better, adapting the technical variation for different acquisition. So the nice thing about developing something that that is generic is that now we can focus on technical variation. It doesn't have to be normal. It doesn't have to be binomial. It actually can be almost anything. So follow the beta binomial exercise. We again, for technical variation, we look at samples that get measured again and again. So these are the mean and variation of one set for intensity data, and these are for phosphoproteomics data. And well, what one sees is that you see the trend of if the mean is getting bigger, and then you get more variation. And as long as your data has a trend, you can model it. And the same with missing data for data-dependent analysis. What happens is that the big one gets sequenced, and the low ones not. So we don't know the identity of the low abundant protein, but the middle one, for some sample you get quantification, but some not, and that's the missing data. And here you can also nicely model the missing data. This is again, see that if this is the frequency of missing data, and we can have a parametric model to model the missing data. I tried this method on a data set that is very well known between epithelial data and non-epithelial data. And you see that for many markers, but this is a two example of the markers that get low p-value in the new test, which should be because these are the markers to differentiate these two groups. And for Lima and the t-test, you get quite high. So, its conclusion, while looking at data is important, we go through what is it, p-value, t-test. For many of you, it's just a reminder. For spectral counting, we have a method to compare this group comparison, also for the pair test, and we end with what we call the S-test that aim to be a very generic method that allow you to study the technical variation of your data. So I have a few extra slides on something that we are working on. So this is protein quantification. We go back to quantification. And what you see here, I don't know if Sandra mentioned this. So one protein, the first thing we do is to digest it into smaller pieces, right? So when we talk about protein quantification, actually we have to aggregate the quantitative data from many different sources. So you get from one protein you have many pieces, many peptides. These peptides are actually come into the machine with a different charge state as well. So this is, for example, one protein, and you can see 29 different peptides. So how do we aggregate the quantitative value of this 29 into a single value to tell people that for this particular sample, this is the amount of the protein, of course, in relative sense. This is an example where you have 8 biological samples, each measured in 3 replicates, and these are the intensity of each observation here. So one data set we are looking at, it's just a very well controlled data set with 12 spiking proteins, so everything is the same, so thousands and thousands of proteins the same. Only these 12 are spiking at different concentration. And well, I think it's a very interesting data, but I think there's a lack of time that you didn't get exposed to this new kind of acquisition. But what you see between the two different kinds of acquisition is that it's very similar, but the II gets a lot more coverage at the lower abundance. But there's one protein that's still very high, wrongly classified using, wrongly quantified using state-of-the-art method of quantification, and that's why we are working on it. So, if you're in AI or machine learning, you should be very familiar with this kind of problem, matrix factorization. You have a matrix X, and it's decomposed into 2 smaller matrices with a smaller rank. And, well, how to do that? You try to minimize this sort of object decomposition. Did everybody here do machine learning? Okay. Well, it is very popular method in recommendation system or matrix completion. So you have missing value and so on. So for protein quantification, actually we're working in log space, and instead of multiplication, we have addition. So it's very interesting to now look at how to deal with missing quantify this single protein from your data. So here's the real data again. We have MS start, it's one method that is quite state-of-the-art, also max LQ. So we have a few state-of-the-art methods, but when it comes to missing data, it's still not to our satisfactory fashion. So we are working on it, and maybe any of you interested in this idea, just come and join us. All right, that's it. I think some of it can be quite heavy. But